Welcome to the Lab to Table podcast, brought to you by Mérieux NutriSciences. In a world where the food industry is constantly evolving, we're here to help you break down the complexities. Join us as we sit down with some of the industry's leading experts to tackle critical food safety updates, decode shifting consumer trends, and deliver the actionable insights you need to stay ahead.
Welcome to the Lab to Table podcast. I am Upasana Hariram, a product manager for analytical microbiology and molecular services here at Mérieux NutriSciences. Today, I'm talking about next generation sequencing with two incredibly smart ladies, Dr. Angela Nguyen, our research director of all things molecular at our Food Science Center, and Dr. Sam Bolton, our Food Safety Programs and Investigations Manager with our Expert Partners Group, which provides consulting and education to food businesses. If you've heard the term NGS and wondered what it actually means and how it works in a real lab, this is the episode for you. But before we dig in, I'd like for you to get to know our guests a little more. My first guest is Dr. Angela Nguyen. She's our research director here at Mérieux NutriSciences, and she's built her career at the intersection of molecular biology and real-world food safety testing. She's even published research on whole genome sequencing methods. Angie, I'd love for you to tell our listeners who you are in your own words and what gets you excited about your work.
Yeah, so thanks for the invitation. My interest has always really been in practical science that really directly impacts public health, which has led me to food microbiology because, after all, everyone has to eat. What gets me really excited about working every day is just the challenge of problem solving. So no 2 days are ever really the same since every issue is distinct. And really, the food safety landscape is always continuously shifting.
I love that. Thanks, Angie. And joining her today is Dr. Sam Bolton. She holds a doctorate degree in food microbiology from Cornell University. She solves daily customer pathogen and spoilage problems and helps customers in developing and strengthening their food safety plans, sanitation standard operating procedures, environmental monitoring programs, and works as a food safety programs and investigations consultant in our expert partners team here at Mérieux. Hi, Sam. Same question for you. Please go ahead and introduce yourselves and what drew you to a career in food safety?
Thank you, Pasana, for the introduction. And as you said, that's basically what my role is at Mérieux NutriSciences. I'm essentially a consultant that helps food companies, address and solve their food safety or food quality problems. And in terms of what drew me to a career in food safety, that's a great question. And I think it actually goes back to what got me interested in science in the first place. So, in my first biology class in high school, we had a project where each group had to design and carry out its own experiment using soybean plants. And most of the groups tested things like, what happens if the plants don't get enough water, or what happens if they don't get enough sunlight. But my group had a different idea, and we wanted to know what happens if you watered plants with beer instead of water. And so amazingly, our teacher allowed us to do this. She bought us a 6-pack of Corona. And for the next couple of months, we watered half of our plants with beer and half of our plants with water. And we really expected the plants that were fed beer to, not do as well as the ones that were fed water. But to everyone's surprise, the beer fed plants actually grew much better. Their root systems became more extensive, and the roots actually started cracking through the red solo cups where they were grown in. And looking back on this as a microbiologist today, I definitely wouldn't recommend beer as a fertilizer or anything. And I'm sure that there were things about that experiment that I do differently today. But that project itself sparked something in me because it was like the first time I experienced the excitement of, asking a question I thought I knew the answer to and scientific evidence proving me wrong, which of course, drove my curiosity into wanting to understand other cases where scientific evidence challenges and shapes what we know about the world around us, particularly with things like plants and food production. So, that led me to, joining a plant sciences research lab as an undergraduate student. And, that lab actually focused on understanding how pathogens like salmonella contaminate and grow in fresh produce like tomatoes and lettuce. And that was my introduction to, food safety research, and everything from there just snowballed, leading me to, my career today, where I, continue to interpret and apply scientific evidence to help develop recommendations for food businesses that, ensure that they're making safe and wholesome food.
Thanks, Sam, for sharing your stories about what got you into food safety. I love it. Let's jump into next gen sequencing. I want to start with a topic that tends to confuse a lot of people, including our own customers that we speak with daily. We often hear NGS treated as a single service, but it's much more of a catch-all term for several different sequencing methods. So, Angie, I was wondering if you could help us unpack that, and what is NGS? What are the different technologies it covers?
Yeah, of course. So NGS actually stands for next generation sequencing, which means really any sequencing technology that is after the what we call traditional or first generation sequencing, which is also known as Sanger sequencing. So NGS is also characterized by the ability to sequence multiple fragments of DNA all at once. So this is known as high throughput sequencing. This includes both short and long read sequencing technologies. So if you want to break it down even further, there are many different applications of NGS technologies, which include things like whole genome sequencing, metabarcoding, and shotgun metagenomics. So whole genome sequencing is sequencing the entire DNA content of a microorganism, like all those A, T, C's, and G's that you hear about that make up the genome. This is typically for single organisms, so to compare samples to each other to understand differences between the DNA of one organism versus another organism. And then you have metabarcoding or metagenomics, which are NGS applications for mixed microbial populations.
Thanks, Angie, for sharing that. So, Sam, when you have to use or I guess propose some of these tools to customers when you're trying to solve their food safety problems, when would you actually choose like a metagenomics versus a whole genome sequencing?
Yeah, so this is a really good question, and it's one that we get quite a bit. So, when you're trying to decide between the two, whether to use whole genome sequencing or metagenomics, there's always a couple of questions that I like to ask. You know, what is the problem that we're trying to solve? What information do we already know? And what information are we missing? Because metagenomics and whole genome sequencing are both very distinct tools in that next generation sequencing toolbox, and they both have they're both very helpful, but they're often used in specialized ways to solve different problems. So, for instance, say my issue is I have, multiple listeria positives in environmental swabs that were taken from my processing facility over the past 2 months. And here, the problem that I want to solve is I want to get rid of that listeria. In this case, I already know that listeria is my problem organism. But the information that I'm missing is where that listeria is coming from. So, to answer the question of where it is coming from, right? To answer that question of what I am missing, I'm reaching for WGS or whole genome sequencing, because whole genome sequencing gives you essentially a high resolution fingerprint of each individual listeria isolate in your facility. And then, from there, we can use that information to determine whether all those listeria positives from, the past 2 months, whether they have the same fingerprint, right? Meaning they're the same strain, or whether they're different. And if they're the same, then that can tell us something about whether we might have an issue with listeria persisting in that facility. And if all the fingerprints are different, that may tell us that it's more likely that listeria is not necessarily persisting in the facility, but is repeatedly coming into the facility. And so, again, that can really help with root cause investigation and things like that. And now, on the other hand, so we talked about, best case uses of WGS. Now, Metagenomics is what I would use when I'm dealing with something like an unexplained spoilage issue. And, I don't necessarily know what's causing the spoilage issue, so I don't actually know the organism, possibly because, my routine microbiological tests, things like testing for lactic acid bacteria, yeast and mold, or coliforms, those are all coming back within spec and normal. So in this case, I'm missing information on what the organism is that's causing my spoilage. And in these types of situations, metagenomics is really useful because it helps us cast a wider net in terms of profiling all the organisms that are in your spoiled product versus all the organisms that are in your non-spoiled product. And from there, you can get some clues into the exact organism that might be causing the spoilage issue, which, in some cases might not even be an organism that could be picked up in routine culture-based testing. So in summary, I like to think of it this way. WGS tells me everything about one organism, including, how it's related to other organisms. And then metagenomics tells you everything about a sample. So including what organisms are in that sample and how abundant those organisms are in relation to each other.
So in metagenomics, you're essentially going in a little blind, and then with whole genome sequencing, um, you need to have the isolate. You need to know the organism. So,
Exactly. No,
okay,
100%. You got it.
thanks. So I know we've heard the terms metagenomics and metabarcoding used interchangeably. So what are the differences between those, Angie? Could you highlight the key differences?
Sure. Yeah, there's a lot of different terms, I guess, in under NGS. So meta barcoding, people also call it targeted metagenomics, if it's not complicated enough. But so meta barcoding or targeted metagenomics and also shotgun metagenomics both fall under the metagenomics umbrella. So both of these things are used to analyze mixed microbial populations, so groups of different microorganisms that are together in one sample, but they look at these groups in different ways. So think of a sample as like a library. So meta barcoding is looking at the library's card catalog. So instead of reading every single book in that library, you're only checking a specific well-known section like the title of each book. So in this approach, you target one specific region of that DNA, which is the ID card of that microorganism. So it's best for identifying who is there. And it's really great for creating like a census of that complex environment. So what bacteria are present in this sample, because it's really cost effective and more efficient at identifying just the organisms that are there. So think of then shotgun metagenomics like reading every single page of every single book in that library. So instead of just checking the titles, you can read or sequence all of the DNA fragments that you find in that library. So you don't just see who is there, but you can see what all that genetic material that is present. And so it's best for understanding not only who is there, but what are they doing. So because you have that full genetic blueprint, you can identify not only the microorganisms that are present, but also their functional capabilities. So for example, you can tell if the bacteria in your food sample has genes that make them resistant to certain antibiotics, or like Sam was mentioning earlier, genes that might cause spoilage defects in a particular product.
Okay, great. This is great. I love the library analogy. Thank you for that. So we focused on some core concepts today, defining exactly what NGS is and outlining some tools that fit into the category of NGS. Angie, you're the leader of our molecular services lab. Could you walk us step by step through what happens to a sample from the moment it arrives in our lab to the moment the customer gets their report?
Yeah, so it really depends on what the customer's goal is, but upon arrival, so the sample enters a NGS workflow that is really tailored to the project's particular goal. The initial part is we homogenize the sample and extract its DNA, and then we check that DNA for both quality and quantity. And then the DNA is prepared for sequencing through a specific protocol that usually incorporates some sort of amplification. And then finally, the sample is ready for sequencing. So the sample goes on the sequencer, and then after that, we have to take that sequencing data and computationally assemble it and analyze that genetic data to address the customer's initial question. And so ultimately, then these findings are then compiled into a detailed report and then delivered to the customer.
A follow-up to that, how long does it take from when you already have a signed contract from a customer? Like, how long does the process take?
Yeah, so the typical process takes about 3 to 4 weeks to do all of the sequencing prep and then analyzing the data and creating a report. But it can also be faster depending on the situation and the need of the customer.
I know Angie and Sam, you'll work together on some of these problems. When does Angie typically bring you in? Is it usually before the process begins, or is it after the customer receives the report?
Sometimes I do get brought in, after a customer receives a report and they're looking more for, understanding and interpreting the findings. But a lot of the times, I think it goes the other way, and I'm bringing clients to Angie, and I think she would agree with me on this. I'm always dropping fires on her. So usually, experts or consultants like myself come into play more towards the beginning when a food business has already done some type of investigation into an issue they're facing on their own. They've done some routine microbiological testing, usually not too extensive or advanced. That can be product testing, that can be environmental testing, but they've ultimately hit a roadblock when we come in, right? They've exhausted their own investigative measures and they need more leads on what could be causing the issue. And so that's usually where I come in, right? Typically, we as consultants are going to start, talking to them and trying to look more closely into their food safety programs to learn, more about what could have caused the issue. But in addition to that, that's when I'll usually come in and recommend something like whole genome sequencing or metagenomics, depending on the situation, so that we can get a better signal for what exactly is either causing the problem or best strategies for solving the problem. So usually I'm sending clients over to Angie's group to get some more advanced molecular sequencing done so that that can be performed. And then once reports come out, that information gets delivered back to me, and then we can, as consultants, better assist that client on, recommendations for how they can address a problem or an issue.
Wonderful. I do want to leave our listeners with something concrete, like a real example of when you'll have used an NGS tool versus traditional testing. Could one of you share a real world use case or a problem that you've solved for a customer? Maybe Sam?
Yeah, I can. So, a good one that comes to mind is an investigation that I worked on a couple years back that involved listeria contamination that kept popping up in a single drain in a food processing facility over several months. And the company that I was working with, initially thought that repeat positives in this single drain meant that listeria was actually living in that drain. And so, they had already, before coming to me, done a lot of significant remediation on, the sanitary condition of that drain. They made it more cleanable, and they started loading that drain up every single night with sanitizer crystals, quat crystals, if you're familiar. But, even after all these interventions, they were still seeing listeria positives in that drain over, a several month period. So, we recommended that this company perform whole genome sequencing on all the listeria isolates, that were obtained from that drain. And from that information, we actually learned that those listeria isolates were not actually related to each other genetically. there was lots of different Listeria strains living in that drain, essentially, or in that drain, essentially. And so this provided us with, an alternative explanation, right? Although the company initially thought that Listeria was the source of, that the drain was the source of Listeria contamination, the WGS data actually helped us determine that the drain was more likely to be the sink that was being repeatedly contaminated with Listeria that was living in, other locations in the facility. And so, further investigation goes on, and we perform what's called a swabathon, where we basically, broke down all the equipment in that room where that drain is to its barest parts and swabbed all of it for Listeria, conducted Listeria testing, and then any positive Listeria samples would go on to have whole genome sequencing conducted on isolates yielded from those positive samples. And so the information that we learned from this swabathon was very, very interesting. So, essentially, as part of our equipment breakdown, we broke down a cooler door that was approximately like 20 feet away from that drain. So we took that door off its hinges, and we saw that at the bottom of that cooler door, it was heavily corroded and rusted, and there was a small hole in that cooler door. And so, we swabbed that hole in that cooler door, and not only was it positive for Listeria, but it was positive for many different strains of Listeria. And so, essentially what that told us was that Listeria was living, in that hole under the cooler door, and was likely repeatedly and that Listeria that was living, in the underside of that cooler door was likely repeatedly going and contaminating the drain during sanitation when, water is being sprayed with high pressure hoses, comes in contact with the cooler door, and then the floor was not really properly sloped. So, water could interact with the cooler door and then flow back into the drain. So, long story short, here, whole genome sequencing helped us better understand that the root cause of Listeria contamination in that drain wasn't actually that the Listeria was living in the drain itself, but it was actually living in a cooler door less than 25 feet away from it. So, whole genome sequencing not only helped us identify the root source of Listeria, but also, helped us better extrapolate root causes, right? As I was talking about, poor sloping of the floor, things like that. And that ultimately helped the company, solve their Listeria issue by implementing appropriate and effective corrective actions.
How I understood it is that it gave a lot of context.
Yes.
Ew. Okay, so that's so interesting. And it was the water that was transporting the Listeria to the drain, right?
Yes. And that's very common. I mean, that's why when we think of drains, we often, they're either the source or the sink if there's a Listeria problem in a drain.
Got it.
Mhm.
Thank you.
Of course.
I'm never going to forget that example. One final question for you, Angie. If a food safety professional is listening to us right now and they've never used NGS, what's the one thing you would want them to take away from today's conversation?
Yeah, I would say that NGS is a really powerful tool that has a wide range of applications. So, although it might seem intimidating at first, like using NGS can often identify a definitive root cause, like Sam was mentioning, when other methods might fall short, which makes it a really valuable tool to have in your toolbox.
Angie, Sam, it's been such a pleasure speaking with you and learning about these tools and these services. This is exactly the kind of conversation our industry needs to learn about these technologies. We'll be back for part two with these ladies, where we're going to talk about the real barriers to NGS adoption and what it's going to take to change that. So don't miss it. Thank you.